Hello friends, welcome back to my YouTube channel Nasrin Padasale. In this video, I'll be sharing you 10 important questions which you can expect for GATE Life Sciences Biochemistry paper. First question is from enzyme inhibition. Slow intravenous infusion of ethanol is a therapy to treat methanol poisoning. The underlying chemical reaction is an example of competitive inhibition. So competitive inhibition occurs when the molecule very similar to the substrate molecule bind to the active site and prevent the binding of actual substrate. So in our case, they say that ethanol is given as a therapy to treat methanol poisoning. So ethanol will be acting as a competitive inhibitor for this enzyme called alcohol dehydrogenase, and they will be competing with the methanol. So they will be occupying all the binding site present in this particular enzyme. So ultimately the concentration of methanol will be reduced. At the same time, we can also stop the conversion of methanol to its toxic metabolite like formate or formaldehyde. Okay, so next I'll be discussing few clinical uses of competitive inhibition. So this you can expect to match the following type of questions. So statin, the target enzyme is HMG-CoA reductase and it is used as an anti-hyperlipidemic agent. Allopurinol, the target enzyme is xanthine oxidase and it is used for the prevention of gout. Methotrexate, the target enzyme is dihydrofolate reductase and it is used in the treatment of cancer, and both these two drugs are being employed for treating high blood pressure, and both have the target enzyme for both the drugs is that angiotensin converting enzyme. The last thing is the discomerol and it is used as an anticoagulant. The target enzyme for this particular competitive inhibitor is vitamin K epoxy reductase. Okay, and next we'll be seeing the example of non-competitive inhibitor, which includes a heavy metal, pepstatin, ethanol and soybean trypsin inhibitor are example of non-competitive inhibitor. Next, we will be seeing therapeutic uses of irreversible inhibitor, which includes disulfiram, where the enzyme inhibitor is aldehyde dehydrogenase and it is used for the treatment of your alcoholism. Next, cyanide ion, the enzyme inhibitor is cytochrome oxidase and it is used for inhibition of your respiratory chain. Fluoride ion, the enzyme inhibitor is enolase and it is used to inhibit glycolysis. Next is malathion and the enzyme inhibitor is acetylcholine esterase and it is used as an organophosphorus insecticide. So why I am discussing these things briefly means you will be definitely expected match the following type of questions from this particular thing which I am discussing in this particular video. So please watch the video continuously. Next is diisopropyl fluorophosphate. The enzyme inhibitor is serine protease and acetylcholine esterase and it is used as a nerve gas. Next, BAL is the enzyme inhibitor is those enzyme in thiol group will be inhibited and it is used as an antidote in case of poisoning due to heavy metal. And the example of the suicide inhibition or suicide inhibitor include your allopurinol, which is used for the treatment of gout, and the enzyme inhibitor is xanthine oxidase, and 5-fluorouracil, which is used in treatment of cancer, and the enzyme inhibitor thymidylate synthase, and aspirin, the enzyme inhibitor cyclooxygenase, and it is used as a non-steroidal anti-inflammatory drug. Next is difluoromethyl ornithine. So the enzyme inhibitor is ornithine decarboxylase and it is used for the treatment of your sleeping sickness. Okay. So the second question is that CH syndrome is caused due to deficiency of your neutrophil and NK cells, because this syndrome is an autosomal recessive disorder and it is associated with the impairment of your neutrophil, macrophage and the natural killer cell. Okay, the identifying features of this disease include progressive neurological dysfunction, and also the person will be having increased tendency to develop leukemia and lymphoma, and there will be depigmentation of hair, skin, eye. The pigment present in the hair, skin and eye will be lost. The third question is that what is the pK value of guanidinium group of arginine? It is 12.5. So here I had given amino group and the functional group and the pK of the respective functional group. So SH group present in cysteine amino acid will have a pK value of 8.3 and OH group present in serine will have a pK value of 13, and same OH group present in threonine will have a pK value of 13, and OH group in tyrosine will have a pK value of 10, and the carboxylic acid group present in aspartic acid will have a pK value of 3.9, and the carboxylic acid present in glutamic acid will be having a pK of 4.3, and the imidazole ring which is present in histidine amino acid will have a pK value of 6, and guanidinium group present in arginine amino acid will have a pK value of 12.5, and NH2 group present in lysine amino acid will have a pK value of 10.5. Next question is fourth question, that is purification of 6-his tagged protein using NINTA, that is nickel nitrotriacetic acid is an example of affinity chromatography. This his tag is also called as polyhistidine tag because it is widely employed as a purification tag for purifying a recombinant protein, and it consists of 6 or more consecutive histidine residues. Okay, the main principle is that these histidine residues will coordinate with a transition metal ion like nickel or cobalt, which will be immobilized in the bead or the resin. So here in affinity chromatography, the resin used is NINTA column. Okay, so those most recombinant protein with histidine tag will be bind to that particular thing. So after that we can elute them with a buffer containing lower pH. Okay, so this particular histidine system is an example of affinity chromatography. It is currently used for purification of recombinant protein. So which of the following glycosidic linkage is present between galactose and glucose that is present in lactose? Okay, so in lactose, glycosidic linkage is present between carbon 1 of galactose and carbon 4 of glucose. Okay, next in sucrose, the glycosidic bond is present between carbon 1 of alpha glucose and carbon 2 of beta fructose. Next in maltose, the glycosidic bond is formed between carbon 1 of one glucose with the carbon 4 of another glucose. Okay, next, which is the most efficient complement fixing class of antibody? The answer is IgM. IgM is having a pentameric structure, and this particular antibody predominates in all your primary immune response to most of the antigen, and it is also a most effective complement fixing immunoglobulin. Okay, so here you can be able to see the properties and biological activities of all the classes and subclasses of the immunoglobulins. So IgM, since it is having a pentameric structure, it have a high molecular weight. Next, normal serum level, the antibody which is present in highest amount is IgG1, which is present 9 mg per ml. The antibody which is present in very low level is IgE, which is present, the level is 0.003 mg per ml. Regarding the in vivo serum half-life, IgG alone having a higher serum half-life, whereas IgE antibody will be having a low serum half-life. That is 2.5 days is the half-life of IgE. And here you can be able to see the antibody which activates the classical complement pathway. So both your IgM as well as IgG are involved in activating a complement pathway, but this IgM is most effective. They have a high tendency to activate the classical complement pathway. Followed by that IgG3, that is a subclass of this particular IgG. At the same time, IgG1 also have the capacity to activate the complement pathway. Next, the antibody which has the capacity to cross placenta is only IgG alone have the capacity. Next, the antibody which is present in the membrane of mature B cell is IgM. Okay. Next, those antibody which have the capacity to bind to Fc receptor phagocytes include your IgG. Next, those antibody which have a respective function called mucosal transport include IgA. Okay. Next, the antibody which induces mast cell degranulation is your IgE antibody. Okay, so IgM is the first isotope produced by the neonate. At the same time, it is the first isotope coming in front of your primary immune response. Next, IgG, IgE, IgD are exist as monomer, whereas the IgA has a capacity to exist at monomer, dimer, trimer, tetramer, and they are membrane bound. IgA is a membrane bound antibody, whereas if you take IgM, it is a monomer, but the secreted form of IgM in serum is a pentamer. Okay. Next, according to Obel hypothesis, inosine at the first position of anticodon cannot form hydrogen bond with guanine. So here you can be able to see this is the green color is a codon, that is your mRNA, and this is a tRNA structure which will be having an anticodon in it. Okay, so here the third position of codon and the first position of anticodon, or else the base that is present in the 3' end of the codon of mRNA and the base that is present in the 5' end of the anticodon has a unique function is that they have a Obel base pairing. Okay, they don't have a usual Watson and Crick base pairing. They will be having a less stringent rules that can be applied. Okay, so here you can be able to see first position of anticodon base and third position of codon base. Say here they are asking about, so if inosine is present in the first position of anticodon, like if it is present here means it has a capacity to base pair with adenine, uracil. At the same time it can also base pair with cytosine. Okay, whereas it can't able to base pair with your guanine. Okay, at the same time, uracil, when it is present in the first position of anticodon base, has a capacity to base pair with adenine. In addition to that, it can also base pair with their guanine. This is under the case called Obel hypothesis. Okay, so if you consider the third base of codon base, so if you consider with the third 3' or the base that is present in the 3' end of the codon of mRNA, that is present in the third position of codon base can be able to base pair with uracil. That's the usual pairing. In addition to that, it can also base pair with inosine. At the same time, when uracil is present in the third position of codon base means, okay, it can pair with guanine. At the same time, it can also pair with inosine. Okay, when cytosine is present in the third position of codon base means it can pair with G, that's a usual base pairing. At the same time, it can also pair with I. Next question is anti-B antibodies are present in the serum of, it is present in the serum of blood group A. Okay, so if you take the blood group A, the antigen present is anti-A and the antigen present in the blood group B is B antigen, and regarding to AB blood group, both anti-A and A antigen and B antigen will be present. Okay, with regarding to antibodies in the plasma, A blood group people will have anti-B antibody in the plasma or serum. Next, B blood group person will be having anti-A antibodies in their serum. These are usual things. This is a basic 12-star level questions, but many of you will be giving a wrong answers for these kind of questions. So, give a brief read about these questions. Okay, whereas O antigen, O group blood group, they don't have any antigen in the membrane of RBC, whereas in the plasma they will be having both anti-A and anti-B. Okay. Next, cell suspension was subjected to membrane disruption followed by differential centrifugation to fractionate the cellular component. Match the centrifugal condition in the column 1 to the appropriate cellular component in column 2. This is simplest question. So, differential centrifugation is a unique centrifugation technique which is employed to separate your cellular organelle, because for some research study they will be working with the cellular organelle, so that's the reason why differential centrifugation will be used. Okay, so it is a repeated centrifugation procedure in a stepwise manner with increasing your RPM. Okay, so first you need to break open the cell and you need to filter the homogenate, and if you centrifuge them at 600 RPM to 1000 RPM for 10 minutes means the nuclei will be sediment in the pellet, because nuclei are the largest subcellular component. So it will be sediment in your pellet, and if you want to isolate some other cellular component means you need to take the supernatant and you need to pour them in a new tube and you need to centrifuge them at 15,000 RPM for 5 minutes. Then after centrifugation you can able to easily isolate your mitochondria, chloroplast, lysosomes and peroxisome. Okay, so after that if you take the supernatant and if you centrifuge them at 1 lakh RPM for 60 minutes, that is 1 hour, you're able to separate your plasma membrane component, microsomal fractions like your fragments of ER, and the large polyribosomes can be segregated in the pellet, so that you can take the pellet, and if you want to work with the ER you can take the pellet. And also if you take the supernatant and if you centrifuge them for 3 lakhs RPM for 2 hours means then you can able to separate your ribosomal unit. At the same time you can able to get your small polyribosomes in the pellet, and the last thing is a soluble part of cytoplasm, that is your cytosol. Okay, so here you can able to see 1000 gravitation force, and for 10 minutes you can isolate your nuclei, and 20,000 gravity, that is 20,000 RPM for 30 minutes you can isolate your lysosomes and peroxisome. Okay, so here you can able to see at 20, that is 15,000 to 20,000 RPM for 5 minutes you can able to isolate your mitochondria, chloroplast, lysosome and peroxisome. Okay, next 80,000 RPM for 1 minute means you can able to isolate microsomes and small vesicles. So here you can able to see at last when you centrifuge at 1,50,000 gravity for 3 hours, then only you can able to isolate ribosomes. Okay. Next question is regarding to, you can definitely expect a question from puromycin. So puromycin is a naturally occurring aminonucleoside antibiotic that will be inhibiting the protein synthesis by a ribosome catalyzed incorporation into carboxy terminal of the elongating Nash enzyme, thereby blocking the further extension, and this puromycin will be resulting in a premature termination of your translation. It is produced by Streptomyces alboniger. Sorry if I pronounce the name incorrectly. Alboniger, which is a gram-positive actinomyce, and this puromycin is a tyrosyl tRNA mimic, and it will be blocking the translation by labeling and releasing the elongated polypeptide chain on the translating ribosome. And it is a unique compound translation reaction because this puromycin itself will be acting as a substrate for this peptidyl transferase enzymatic reaction. This puromycin will be mimicking the 3' adenosine of your tRNA structure charged with modified tyrosine. That's why it is called as a tyrosyl tRNA mimic. Okay, so if you check the structure of your puromycin as well as tyrosine, both will be having a similar structure. The only difference is regarding to the nature of bond that is present between the amino acid and the ribosome. In tyrosyl RNA, you will be having a ester bond, and in a puromycin, you will be having a peptide bond. So since both are having a similar structure, in the place of the tyrosine functioning tRNA, this puromycin will be entered into the P site. Ultimately, if the puromycin has been bind means, then further process of translation will be stopped, so that the elongation of the polypeptide chain will be stopped, and it will be producing a truncated or non-functional protein. So this puromycin blocks the translation both in your prokaryotes as well as in eukaryotes. Okay, so that's the activity of puromycin. So it is isolated from this particular actinomides. Okay, so these are valid important points. So the reaction catalyzed by this procedure is called as puromycylation, and it is an energy independent process. Once this particular puromycin has been incorporated means that the entire ribosomal subunit will be disassembled, and it will be releasing the peptidyl puromycin product will be released. Okay, so currently this puromycin is also used for few labeling studies. Also, this puromycin is being employed. These are the important questions that you should study. You can expect these questions for your GATE biochemistry paper. So if it is fine, useful, please subscribe to my YouTube channel. Thank you friends, thanks for watching this video.